Hey, welcome back everyone. It is finally time for us to start talking about what goes on during photosynthesis. Now, this video in particular is actually going to be a little on the short side because it's actually kind of interesting. A lot of what we're going to see here during the light-dependent reactions is very similar to a lot of what we saw during the electron transport chain in cellular respiration. So, since you have presumably already watched those videos, I'm not going to belabor those points, and we're mostly just going to focus on the new stuff here. Okay, so we are going to talk about what are called the non-cyclic light reactions, which are going to essentially make for us two high-energy molecules, ATP and NADPH. The cyclic light reactions, which we are not going to talk about, just make ATP. So we definitely need the non-cyclic light reactions because both ATP and NADPH are necessary. The light reactions are going to utilize photosystems, which are essentially just big complexes of both pigment and protein, and reaction centers that are embedded within the thylakoid membrane. So this is where the chlorophyll pigment comes together with the proteins that the cell makes, and they together are going to absorb light and eventually use that to make energy for us. So these photosystems are very much like antennae. They are there to collect energy from the sun, and they will use that energy to excite electrons that came from water, as you will recall, and those high-energy electrons will eventually be donated to the electron carrier NADPH and or used to synthesize ATP. There are two photosystems. They are called Photosystem 2 and Photosystem 1. Now, you might be wondering, well, why on earth are you mentioning Photosystem 2 first? Well, the reason I'm mentioning Photosystem 2 first is because, chronologically, it actually is the first one to act. Well, why is it called Photosystem 2 then? Well, it actually has to do with in which order these photosystems were discovered by scientists. It just so happens that Photosystem 2, which is the photosystem that acts first, it was actually discovered second, so that's why it's called Photosystem 2 rather than Photosystem 1. So this is just a little curiosity that you're going to have to kind of get used to, the fact that something that is labeled 2 actually goes first and something that is labeled 1 goes second. So I don't know what to tell you about that other than what I just did. So photosystem 2 is going to receive electrons from the splitting of water into oxygen. So water, again, is acting as our electron donor. Light energy is absorbed by the pigment that is in the photosystem, and then that energy is transferred to those electrons. They become excited, they take on a lot of energy, and then those electrons are passed through an electron transport chain in the thylakoid membrane, and we are going to use the hydrogen ion gradient that we make to synthesize ATP by chemiosmosis. Again, a concept we are already very familiar with. Now, once these electrons pass through this electron transport chain, if this were cellular respiration, we would expect those electrons to be accepted by oxygen, and then that would be the end of the line. Well, once we have leached all the energy out of these electrons that came from water, once they pass through this electron transport chain, those electrons that now have low energy, they are then going to complex with photosystem 1. So photosystem 1 will take those now low energy electrons and excite them again by absorbing more light. So in photosystem 2, we energize these electrons, we transfer as much of that energy as we can into a chemiosmotic gradient, and then we take those same electrons and we re-energize them by giving them more light. So we re-energize those electrons with energy absorption. These high-energy electrons are then going to be passed on to NADP plus to form NADPH. So, in the grand scheme of things, between photosystem 2 and photosystem 1, we are going to make both NADPH and ATP, which are both going to be necessary for the Calvin cycle / dark reactions. Here you can see the actions of both photosystem 2 and photosystem 1. As we said, our electron donor is going to be a molecule of water. We donate these electrons to the pigment that are complexed together with the proteins in photosystem 2. The electrons are energized by absorbed energy from the sun, and then we pass those electrons through an electron transport chain, which, as we've seen before, is going to be used to make ATP by chemiosmosis. Those electrons, once we've leached all the energy out of them, are going to complex with the reaction center in photosystem 1, we're going to absorb more light from the sun and re-energize those electrons. And those electrons, rather than going through a whole nother electron transport chain, those electrons are going to be passed on to NADP to produce a high-energy NADPH molecule, both of which, the NADPH and the ATP, are necessary for the Calvin cycle reactions. So here you can actually see, if you don't quite remember everything from cellular respiration, we'll give you an idea of everything that's happening here, including the electron transport chain. So just like we saw in mitochondria, this is going to consist of cytochrome complexes and iron-containing proteins. One we haven't seen before called Plastoquinone, but that's just another mobile electron carrier that serves a very, very similar function to the ubiquinone that we saw in the mitochondria. So here you can see photosystem 2. It's going to take on electrons that are donated from water. We're going to absorb light and energize those electrons, then pass it through this electron transport chain. And in the process, we are going to pump hydrogen ions from the stroma into the thylakoid space inside of the thylakoid. So very similar to what we did in the mitochondria, in which we pumped hydrogen ions from the matrix into the inner membrane space. So as these high-energy electrons get passed along through the electron transport chain, we are going to pump hydrogen ions and produce a very large hydrogen ion gradient in the thylakoid space. And then ATP synthase is going to use this hydrogen gradient to synthesize ATP by chemiosmosis. So that's everything for the first half of the light reactions. Once those high-energy electrons have been spent, they're going to be taken on by photosystem 1, which will absorb more light energy, and then these electrons are going to be used by an enzyme called NADP reductase to turn NADP plus into NADPH. So as you can see here, the fortuitous thing about this is that photosystem 1 is directly adjacent to that enzyme NADP reductase that allows for the direct transfer of those high-energy electrons from photosystem 1 onto NADP to produce our NADPH. So it's also important to recognize that the hydrogen gradient that you see accumulating in the thylakoid space, it doesn't just come from the pumping in the electron transport chain. Every time water donates electrons to photosystem 2, it's going to produce a molecule of oxygen and then 4 hydrogen ions as well. So 2 water molecules, 4 hydrogens, and 1 oxygen molecule. Okay, and like I said, it's a short video. That is essentially everything that's going on during the light reactions. The thing for you to focus on, and I don't want you to get complacent because that was a very short video, the thing to focus on here is to make sure that you're keeping your eye on the 2 goals of the light-dependent reactions. Make ATP, make NADPH. Photosystem 2 produces a chemiosmotic gradient that we use to make ATP, very similar to what we did in cellular respiration. And then photosystem 1 comes along and re-energizes those electrons and then transfers them right onto NADP to make NADPH. So the whole purpose of this is that both of those high-energy molecules are going to be absolutely necessary for what we're going to do next during the Calvin cycle. Alright, so that's going to do it for this video. Next video, as you can imagine, is going to be a little bit longer, but I hope you'll look forward to it, and I will see you next time.